This video will describe the processes involved in the data collection and website resource production of the Allen Human Brain Atlases. We will begin with the Adult Human Brain Atlas at human.brain-map.org. This information is also available from the Nature article published in 2012 that describes the methodology used to capture gene expression across the entire human brain. This figure, taken from the article just mentioned, describes the processes involved in building this resource. Specimen acquisition was more problematic than expected, given that the Allen Institute wanted to ensure the RNA integrity of tissue samples collected. Donor brains were imaged via magnetic resonance, slabbed, frozen, and sent to the Allen Institute within 30 hours post-mortem. Getting consent, given they were looking for healthy whole brains under that time frame, was not easy, and the resultant resource contains only one of the intended 5 female brains, although it does contain 5 of the intended 5 male brains. I'll walk through these steps undertaken to collect the various data modalities in the next section of this video. Data collection, informatics and data analysis, data integration, storage and retrieval were all automated processes and enable the construction of the web interface that utilizes unique visualizations to combine brain structure and gene expression. This atlas includes genome-wide transcriptomic information that is mapped to 2- and 3-dimensional reference spaces. It also includes cellular resolution images taken by in situ hybridization for a select number of genes across select structures. Let's take a look at the processes involved in creating this gene expression map of the human brain, starting with acquisition of the tissue and how it was processed. Donors were between the ages of 18 and 68 with no known clinically relevant conditions. A strict protocol was developed to ensure that the brains were of high quality and yet clinically unremarkable. More information on the selection of donors and the processing of the tissues can be found under the Documentation tab of the Human Brain Atlas. Depending on the brain bank where the brains were acquired, magnetic resonance images were taken either in cranio or ex cranio. These images show how the brains were imaged while they were out of the skull. Slabbing, freezing, and sectioning of whole brains were processes that required development and refinement. Once the slabs were received by the Allen Institute, an initial 6x8 coronal section was collected and Nissl stained, primarily to determine a baseline location for each slab. Each slab was cut into blocks that would fit on 2x3-inch slides, and histology in the form of Nissl stains, immunohistochemistry, and in situ hybridization was performed primarily to capture the cytoarchitecture to understand the location in the brain. The final millimeter of each slab was reserved for RNA extraction, either manual dissection of cortical samples as pictured here, or laser microdissection for small or odd-shaped brain structures, such as in the subcortex. Gene expression was detected using microarray analysis, in which RNA was extracted from the macro or microdissected samples, labeled cDNA was created from the extracted RNA, and loaded onto Agilent chip arrays. The resultant quantitative measure of gene expression for over 60,000 probes was manually mapped into the brain space as detected by MRI. The web interface includes approximately 500 samples per hemisphere of 8 distinct brain hemispheres. As well as the neurotransmitter study mentioned earlier that was conducted as part of the whole brain survey in the Human Brain Atlas, in situ hybridization data has been collected from human brain specimens for over a decade at the Allen Institute. These studies, which can be found in the Human Brain Atlas at human.brain-map.org, include 55 genes across subcortical regions of a male and female donor, a 1,000 gene survey which was conducted in 2 regions of the brain, the visual and the temporal cortex. These 1,000 genes sampled were selected based on the top 1,000 genes searched for in the Allen Mouse Brain Atlas. 2 diseased studies were also conducted, a schizophrenia study of 60 genes in the dorsolateral prefrontal cortex, and an autism study in which 25 genes from various cortical regions was probed for in 11 autism and 11 control cases. The Allen Institute was also part of a consortium that performed a multimodal study to understand gene expression over development of the human brain. This study examines gene expression over the human lifespan and includes donor brains that range from 8 weeks post-conception through to adult brains in the 5th decade. This dataset measured gene expression using RNA sequencing in only 16 structures, so the anatomic resolution of this atlas is rather coarse, although the temporal and transcriptome level resolution is very high. Two-dimensional reference atlases, both prenatal and adult, were created for this resource. This slide illustrates the structures that were collected for RNA sequencing at the various developmental stages. Note the limited structural coverage. This slide also shows the breakdown of donors across development, with an indicator of the sex of the donors at the available stages. RNA sequencing was not the only data modality collected in this study, although, along with ISH images, it is the only one visualized through the web portal at brainspan.org. To access the other modalities, such as MRI and DTI, methylation, and microRNA, see the Download tab from the brainspan.org website. The last thing you need to know that because this data set includes human sequences, the data you can download is normalized data that are used in the heat maps, so mismatched data is not available in this resource. This overview should give you enough information to take advantage of the data available in these resources.